All right. So the first thing that we are going to do, by the way, beautiful morning, went on a run, went on a walk, thought about biology. All right. Let's get into it. So I'm wondering if I just want to start totally from scratch. And I think that that might be the right idea because, all right. So let's just start from scratch. We're going to make a new folder. Or I'll just close all this and we're going to start with just puck19.gb. All right. Yeah, let's make a new folder. Try 2. Okay, we're gonna open a folder. I think if I close wait, actually, isn't there? Hold on. Okay. I should be able to open folder as project. Try 2. Okay, open. Okay, cool. So what I'm gonna do is I'm actually gonna copy only. Actually, you know what? I'm not gonna copy anything. We're gonna start totally from scratch. Okay, so let's get some context on what really is going on here. So let me pull up Firefox and I'll probably need to charge this. Oh yeah, this thing's like dead. Okay, hold on. Shit. Okay, so I don't think I can use this pen while it's plugged in, can I? No. Okay. That's just great. Um um that's so dumb. Whatever. We'll plug it in like 5 minutes. Does this still have charge though? Okay, yes it does. The hell? Okay, there we go. All right. So, damn it. Okay. All right. So, um okay, bio. Oh my gosh. I am all over the place today. Okay. All right, bioengineering So we're basically starting from scratch of bioengineering. And what does that actually mean? So to me, basically, I was thinking about what does that mean? Well, it means like things like making cells glow as like a or like editing the genome to make this is sort of a more generic version of the previous thing. Make life do new, or let's say living things new, and maybe new stuff. Okay, that's like kind of the Okay, well, it's good to know that OBS can recover when I accidentally go into airplane mode. All right, so when I basically joined this place, BioCurious, it's basically a lab. It's like a gym membership. You pay and you can use their stuff. Someone handed me a petri dish, and on it were two things: a strain of E. coli called NEB stable, and another strain, NEB stable, with a plasmid called P glow. And for anyone that doesn't know, plasmid has a gene on it. There's other things that it does, but this gene, this is a circular piece of DNA. Okay, and if you put this thing into a cell, this cell will start doing all the things to basically make this gene do something, because this gene, codes for glowing, and rather we say fluorescent protein. Okay, so that's kind of like where we started out. But now, someone just gave me this basically glowing cell that I could propagate and make glow and look at. Okay, but what is the next step? After glowing cells, what's the next? Okay, and this question led me to something called Gibson assembly. And what I'll describe that as is basically a way to stitch pieces of DNA together. So if we have a plasmid, well, actually, let's back up. If I have a piece of DNA like this and another piece of DNA like this, and this part is common, then we have some reagent that has enzymes in it that will basically, stitch these together so that we have one big long one. So, if this is DNA B and this is A, then we have AB. Okay, now that's pretty cool, but how do we make it a plasmid? Or how do we, rather, this is sort of the common workflow, is you take a plasmid and you add a gene of interest. Okay, and then you get Okay. So, but there's a number of complications. So this might seem maybe simple on the surface, but there's some complications. First off, if we're starting with a plasmid, and usually what we call this is like a vector backbone, and all that means is like it's a plasmid that is designed for the purpose of putting something else into it. So it's kind of like an engineered plasmid that has the designed purpose of using it to put into cells to express your gene of interest. And the one we've settled on is called Puck19. One, because someone else had it in my lab, and it's relatively small. And the main thing is that I don't have to buy it. I already have it, and I have it in what's called. I have it in cells, so I don't actually have it in pure form. So that leads us to kind of the first step of this whole procedure, which is purify the plasmid from the cells. You'll typically see this called a mini prep. Okay. And that's like the first step. But the second step is going to be actually called linearizing this. And, you know, in the sort of big tech tree of all this, this ends up being a pretty interesting complication because it leads you to having to do a bunch of you know, some of the big famous things that you'll hear about when people talk about molecular biology, and that is basically PCR. Okay, but what PCR is going to do for us is it's going to take this circle and turn it into a line. And the reason we need to do that is that the way this Gibson assembly thing works is that they both need to be linear. And actually, what we, you know, yeah, so this picture is good. Is the overall thing that we want to do is this. We have a plasmid, and then we have a gene of interest, which is ordered online. And actually, the manufacturing of this is something that we definitely want to go into. But you can, if you want a phrase, you can look up phosphorimidite chemistry. This is how they make it, and they make it through this. It's another one of these like iterated chemical reactions, just like PCR, where you can progressively add individual nucleotides onto the end of a growing, what they call is oligo, and we'll get into that in a second. Oligonucleotide, it's a word, and that's all I'll say about that. It's a word. I forget what oligo's root etymologically is, but that's sufficient for what we know. Okay, so let's get into linearization, though. Linearization is basically going to work through PCR. Now, the whirlwind thing of PCR is, let's say we have a piece of. Okay, let's just do PCR. So we'll do this here. Okay, PCR. Okay, so let's say that we have a piece of DNA. Okay, and we actually want a bunch of copies of this thing. We'll call this G for gene. Okay, well, one thing that you need to know, and this I basically the only way that you're gonna actually get this 5' and 3' thing is, first off, go look up basically the actual molecular structure of DNA, and you'll understand this 5' and 3' thing. But basically, all it means is that DNA is composed of two strands that are going in opposite directions. And this is really important because it turns out that there's an enzyme called DNA polymerase. And what DNA polymerase does is let's say that you had a piece of DNA like this. And we can do do this. So this is G. And let's say that you had a little starting bit here. Let's say that you had this thing. Okay, so this is going to be a 5 and 3. And this will be a 3 this way. And let's say we had a starting bit that's bound to this. So I guess one thing that you would want to know is A binds with T and C binds with G. Some of this stuff I'm just going to necessarily have to skip over because it's so elementary that if you don't know that then I don't know what to tell you. But anyways, what DNA polymerase is going to do is it's going to come on here, bind to this thing, and start adding the bases onto the end so that this thing ends up filling up. So then what you'll, but it doesn't know when to stop, so it's going to keep going to the end here. But then what's going to happen is we heat this up and denature it, heat it up, and now this is going to split apart, and so now we have our original strand, and we can say that this is the G zone. Okay, and we went farther, but since we had this starting point, because the thing is, polymerase is only gonna add on to the thing, so it needs this little starting point. If the starting point of this, and we call this thing the oligo or primer, or whatever. Let's see, is that gonna be too small? Might be too small. Anyways, the next time, let's say we have another primer and this one goes this way. Well, then this is going to go to the end here, but it's going to stop here because there's no more DNA at this point. No more DNA because it needs that other strand. It can't just add on, because the way it works is by basically reading off the template strand and adding bases that are complementary to it. Okay, so the idea now is that we come up with primers that instead of amplifying a small g here, we amplify everything else. So we amplify everything else. And then since it's broken at the ends, it turns into a line. Just like this, even if we had a circular piece of DNA and we were trying to amplify a little gene on here, and we had our primers going this way, and this got amplified, it would still end up being a line. Still be linear DNA. Okay, so basically the idea is we just make primers that point outward. Okay, so this leads us into a sort of a subsection of a goal is now we need to design the primers. Because primers, basically you want to make it energetically favorable. I guess one thing that I left out here is that once you have this, you basically do this whole process of denature, anneal, and elongate x 30 times or so. And then it'll basically exponentially amplify the sequence of interest. So you have a ton of copies. Okay, but designing these primers is actually something that has a good amount of tooling, software tooling, already in existence. So one tool that we found is called Primer 3. And it has, or the Python version of it, which is just a wrapper script around the C library. And what this does is it allows you to specify a whole bunch of settings, a cost model on like kind of what you're looking for, because primers are usually what do they say, like 18 to 40 base pairs long, and you want to have something that has an even mix of A's and T's versus C's and G's, and that's due to the, basically my understanding is the number of hydrogen bonds between pyrimidines and purines. One has 3 and the other has 2. So basically this number that they call TM I think the M is for like melt or something. I don't actually know what it is, but it's not the melting temperature. What that's like a misconception I had. What TM is basically the temperature when the template and the um primer, or rather when the primer template temperature when the primer 50% of the primers have annealed to template. So that's all good. Now, let me just check my volume because I think I was a little loud last time. And anyways, the goal is that if you have so let's do this 5 3 3 5. So the section that binds to here, we usually call this the reverse primer. We usually call this because, oh, I guess, did I say this? That polymerase only adds onto the 3' end. So that enzyme is just only able to add onto the 3'. So basically, when we're looking at trying to design the primers, we need to think about that directionality. So back in here, we're going to have a 5 and a 3 and a 5 and a 3. So then we can go like this. Cool. So, and this is usually called the forward primer. But now what we're going to do for our inverse PCR is we are actually going to basically move this whole thing over here so that there's a junction that separates so that we're going to end up looping around this way, as opposed to doing something pointing towards each other, they point away from each other. So that's kind of the idea there. Okay, so we had ChatGPT generated. So we generated a script to look for a pair of primers. And one thing that I guess we should look at is, let's do okay. So now we're at the point where when we're going to talk about primer design, we need to actually look at the structure of Puck 19. So let's do that. Okay, so I'm just going to draw a circle here. Okay. Puck 19 has a couple of things on it. I'm only going to talk about a couple. The first is called this thing called the origin of replication. Now, I am not even close to an expert, or do I even really understand what the origin, the mechanisms of DNA replication in prokaryotes is. This is something that I got my essential cell biology next to me, and I'm basically just trying to wrap up this project so I can start to read it and get more into the actual, understanding what is known about bacteria and life in general. Okay, but that's the first feature. This basically is a DNA marker that bacteria have enzymes for that recognize it, that basically, if the cell is dividing, sort of has this enzyme that looks at and finds origins of replication to initiate DNA replication when the cell is going to divide. So this is how we can then produce two daughter cells that have copies of not only the chromosomal DNA of the bacteria, but also this plasmid that's in there. Because plasmids are actually not just a piece of engineering, but also something that bacteria evidently evolved to transmit and share with other bacteria in the environment. Okay, so that's the first feature that we wanted to look at. The second feature is going to be, and I don't know if this is the right order, but the order does not really matter. And usually you'll see this going this way. And what a feature labeled with a clockwise arrow means is that it's usually on the, well, I don't know. It's on the forward strand. It's on the top strand usually, because you'll typically see it. Well, we'll get to it. We'll get to it. But I really like this idea. Like the streams, we just sort of. I used to do this with Cast 3. It's like you just start from scratch every day, and then it becomes so second nature that it's like whatever. All right. So the second thing is going to be this amp R, and this basically gives resistance to ampicillin. Gives resistance to host cell. So basically, they call this a selection marker because what it allows us to do is if we end up putting this plasmid in our bacteria and then we spray a bunch of ampicillin on it, well, any of the bacteria that didn't get the plasmid shouldn't have the protein that allows them to be resistant to the antibiotic and therefore will die. So it allows us to select for only the bacteria that have the plasmid in it. Okay, then there's another piece in here, and this is probably the most important piece in here. This is going to be, and I guess we'll say this, it's like 2,800 base pairs. So we'll point this out too. So this is going to be the lac promoter, and then this is lac Z alpha. Okay, so and I don't fully understand okay, well I'll explain what we know. So basically a gene typically has a promoter, and then it has a ribosome binding site. I don't really like that those here, what I'll do is primer, promoter, RBS, and there's spacers in between here, and these spacers are like kind of random. And then we have the coding sequence. So this is actually like the amino acid codon list. All right, and then a terminator. So this is kind of a gene. Now, basically a promoter is a thing, and let's actually just look up gene promoter. How do they define it? Yeah, it's a sequence of DNA to which proteins bind to initiate transcription. So I guess I'm leaving out some stuff. So let's just go over the central dogma of biology. We have DNA. Okay, DNA gets transcribed by RNA polymerase, okay, into mRNA. And this is kind of, we don't really need that. Okay, which gets translated in ribosomes, which interestingly are made of RNA a lot into proteins, and proteins are kind of the molecules of action. They kind of do all of the cool stuff and make all the interesting chemistry happen. So anyways, a promoter is basically a place that RNAP can recognize and sort of attach to, and then start transcription and make some mRNA. Okay, so that's a central dogma, and that's sort of okay. So that's the promoter, and so lac, uh, uh, they call this the lac promoter because it was found originally in this mechanism that I don't know what host cell originally had it, but basically there is there were a series of genes found together that basically enabled the metabolism and sort of packaging or stuff like that of lactose, the sugar, and the cleavage of it. And so basically the idea was that this promoter had an interesting switch on it, because if there's no lactose in the environment, we don't want to be producing all of these genes to metabolize lactose, because it's not going to work. You're not going to get any benefit unless you actually have lactose in the environment. So basically this thing has an inhibitor. So there's this like lac I that binds to this. Or really it's in the promoter. In the promoter, the lac I binds. And it blocks RNA polymerase from starting transcription because it just can't physically fit in there. And I think we could probably just switch over to this diagram to look at this. So this is the lac operon. They call it an operon because they don't call it just the lac gene, because there's actually multiple proteins that are coded for in a single little package, and it's basically just a compression mechanism by evolution, because it's like, all right, we recognize that these proteins basically always need to turn on at the same time. Why don't we just put them on the same little strip? So 4 is, they call it the operator, but it's basically the place that the inhibitor can bind to. So that's that lac inhibitor, and RNA polymerase binds to it. So where were we going with this? Because this kind of went into the weeds. So okay, we're still talking about primer design. So anyways, LAC-ZA just happens to be one of these lac genes that they put in PUC19, and it turns out that this can be used for like blue white screening. Now I don't really care about that. And so actually what I ended up doing is I had my search regions. So I had my search region be basically so I picked basically these two search regions with the intention that this one would point this way and this one would point this way, so that then I would basically cut out this whole lac nonsense because I don't need it. I'm trying to put my own gene in it. Oh, and I guess one thing that we'll say is, after glowing cells, what's the next step? A different color. Of course it is. A different color. It's so simple. What about a different color? And it's funny because I like that, because it's a simple question. Why don't we just pick a different color? And it leads you down this whole rabbit hole that like, you know, you maybe weren't expecting, which is the cool part of this whole thing. Okay, so we searched there in primer three, this program. Or actually, wait, let's do this. Let's do this up here. Okay, so primer 3 gave us a bunch of options with stats on that TM stuff, the length, the G, they call it the GC percentage, and then we can use that to make a decision. So so we picked the first one. Okay, let's pick the first one. So basically, this is like the kind of the first thing we need to buy in doing this whole thing. You physically, you go to a website and you just write down these letters and then you buy it. And the place that I'm gonna buy them from is called IDT. Okay, so that's pretty cool. But after we linearize it, we need this other part, right? We need this gene fragment here, gene of interest. So the other thing that we need to add is we need to. Okay. All right. Let's just say. So now that we have the primers, so now we have the primers. We do PCR, which we went in detail. We went in we went in like a moderate amount of detail explaining it, but there are many better videos on that. To get the linear puck 19 without the lac gene stuff. So we got rid of that. So now we're gonna have something like this that has like Ori. Well, actually, depending on where we cut it, it's kind of not gonna be like that. It's gonna be like let's see. Since we're cutting it here, we're gonna have like. Actually, that's right. We're gonna have like this Ori AmpR. And we want to keep AmpR because we actually do want a selection marker. And so now our goal in here is to add this bit that loops this around that makes it so this part is the gene of interest that makes it glow. So how do we do that? Well, we buy it. That's kind of the most unsatisfying thing about this whole process to me is that we basically, there's this technology and it's not commercial, it's not so democratized that you can just get the machine to print the DNA. You basically have to go to these services that print the DNA. But again, it's based on this phosphorimidite chemistry, which I don't know in depth, but I am planning on in the future doing some videos going real in depth in phosphorimidite chemistry because it's like that is such an enabling technology that's probably worth knowing. All right. So what do? We order it from a company called Twist. Okay. So this will be, they call it M Cherry. Cherry because it glows like a red like a cherry. Okay, so now we can go back to the original thing, which was this Gibson assembly thing. And basically the only thing that we need to remember is that when we design, so if twist was a function, it takes basically a sequence and gives you actual DNA. So text sequence, actual DNA fragment. And one thing that's interesting to me is they give you maybe about 750 nanograms of DNA, which is not that much. But if you calculate based on the molecular weight, it's actually a lot of pieces of DNA. So mcherry, for context alone, no promoter, is about 711 base pairs. And they price this thing. This needs to have the size of like 200 to they're 0.2 to 5 kbp or something like that. So kilobase pairs for anybody that. All right, but then we need to add. We need a promoter. So we're basically in the segment of, wait. We need to design our gene. So we need a promoter. So how do we do that? How we could just, oh, and one thing that I sort of glossed over is this lac I thing is a cool feature. Cool. So when we say we need a promoter, how do we want the gene to behave? Do we want it to always be glowing? Having it always be glowing would be fine. There's not really an issue with that. But it's cooler to have a switch where we can turn it on. Okay, so we want to be able to turn on gene expression. We call this inducing. So we could then, okay, so the easiest option, use the lac promoter again. Well, it turns out some badasses in the 1980s, oops, Genentech scientists made a better version. Better version. You can figure out what that means by reading the paper, but needless to say, it's better. Called TAC. So this is called P-TAC. Or rather, P-TAC-I. But they have two versions of it, but we're only going to use one. All right, so this is how we found the promoter. And so that's the promoter we're going to use. So this is actually what we're going to use. So now, and then how do we get the coding sequence? The coding sequence came from FP Base, a website that basically has the amino acid database of data about fluorescent proteins. So we got the AA sequence from AA. And then what did we do? We took the amino acid sequence and we gave it to Twist. Gave to Twist, which converted it to DNA, given that we plan to express the protein in E. coli. So it turns out that the amino acids that life tends to use in different species is different. As they evolved, they sort of changed which amino acids they like to use. At least this is my understanding. And in doing so, then when we want to convert an amino acid sequence back to DNA, because it's not a one-to-one mapping, we need to basically take into account which species we plan on expressing it in, and then coming up with the amino acid sequence or codons that is most likely to work. And so we just use Twist's online tool to do that. Okay, once we had that, now we need to do the overlaps. Overlaps is sort of the last step because okay, at this stage, then we have the promoter, and then the promoter. I also, let's see, what did I do for the Oh, where did we? Oh, what we did is we searched on AdGene for plasmids that had the, I think it was the lac operator sequence. I forget, and copied that. Wow, that's terrible. So that's how we got this promoter operator RBS, and then the coding sequence from Twist. So this is from AdGene. Okay, so now the only step left is this overhangs. So these, if you remember, are required in doing this thing here. So for Gibson assembly, you need to have the overhangs, and you actually need to have the overhangs on both sides so that it turns from a line into a circle, because it needs to attach on both sides. So then we went through an extensive thing to figure out that what those overhangs needed to be. Honestly, it's a lot simpler than I made it out to be, but sometimes that's how it is. I'll keep this in line here. So this is from Twist. Okay, so that is kind of a recap. Now we're actually going to go through the process of doing all this. So, okay, wait. So how do we find the overhangs? So how do we compute the overhangs? How to compute overhangs. So I'm probably not going to go into this because I'm ready to just start working now, but let's see, how long is our review session? Because I don't want the review to be more than an hour. So, but basically, how do you find the overhangs? Well, there's sort of two ways. First, you can take, so if we call this sequence G, then you can actually do PCR. Let's say you have G here. If you add the primer like this with an overhang, so this is primer reverse, and then this is, if you have this overhang, if you do some arrow chasing, you'll see that you can actually add bases and you get the whole thing with the overhang. So you can add overhangs with basically with long primers. But it turns out that I don't really want to do that because I can just, because one it requires another PCR step, and PCR can take a while, and I don't know. This is already a big project with a lot of moving parts for me, so I'm trying to minimize the difficulty. So what we're doing is we're doing a little bit of extra design work ahead of time to basically just order the whole thing from Twist. Okay, so that is what we're going to do right now, is we are basically going to, in software, basically design this experiment. All right, so we're good. All right, so the first step here is we're going to go to add gene, and we're going to look up our backbone, PUC19, okay? We're going to go to plasmids. Here we go. We got PUC19. What does this flame mean? So it's very popular. Okay, so we're going to download this thing. Sequences. So one thing, I used to download the GenBank, but now I'm going to download the Snap Gene. And I guess I already did that. Okay, we're going to cut this out and put it into sequences. Try 2. All right, so now we have PUC19. And we want to close this. Oh, wait. Let's close this. Okay, so here we go. This is what PUC19 looks like. And remember, in my drawing, we can check how close my drawing was to this this whole thing. So we have the origin of replication. We have this lac ZA thing, and then there's a whole bunch of other stuff in here, which is like, here's the lac promoter. That's important. Here's the lac operator that we talked about. All of these things here, these like names, I actually don't really like them because they're called restriction sites, or basically there's a special class of proteins or enzymes that cut DNA in specific places. And we end up actually having to use that. Well, I don't know if I have to. I'm kind of suspicious on whether or not it's absolutely necessary, but I am going to do it because I think one of my lab mates has the reagents. All right, but here are the 3 main features that we talked about. We have this lac part, we have the origin of replication, and then we have this ampicillin resistance thing. Okay. So now one thing that I want to do is I want to look up, basically, we're going to look up online Snap Gene lac operator. Or actually, you know what? Let's do TAC promoter. TAC promoter. So basically this tach promoter, there's like this -35 region and this -10 region, and what these are is they're basically little 6-segment DNA things that are that allow RNA polymerase to bind to them. That's basically it. So what we're gonna do here is take this sequence, and I'm actually going to just search by sequence. And I think maybe this is not going to work because of the spaces. Okay, I guess it's fine. Now, actually, what did I download? I kind of want to look at my downloads because I want to make sure that I actually am not changing this up. $177,907. Okay, so this is what I used. So what I did here then is and you can actually buy these, which is kind of cool. But we're gonna download the snap gene, and here I'm gonna prepend this thing to it, and we're gonna go. It's this one. Put this here. Okay, we're gonna cut it out. Bio, sequences, try to. Okay, so we now have almost everything. We now need to go to Twist Biosciences. And I feel like there's probably a better way to do this. Actually, let me do this chat. What's the best way to take an amino acid sequence from FP base and convert it into nucleotides? I plan on expressing in NAB stable E. coli. Because what we've been doing, and while we just wait on that thing, basically what we've been doing is we go to FP base, we look up M cherry, then we take this thing, we copy it, we go to Twist Biosciences, I guess. You know, interestingly, actually, I feel like Snapgene probably has it. New protein file. Let's create a sequence. We do this. And then can I put a star at the end, or does that not work? Actions. Reverse translate. Generic code. Okay, so we do have it in here, but now we're looking at codon usage table. So it would be interesting to actually so what did they say? Bacterial. Wait, no. Wait. What did they say? Bacterial. Okay. So see, the interesting thing here is that this is this is 711, but it's using TAA. So wait, let's so actually I want to rename this. Can I rename this? I guess it's not saved yet. Oh, wait, save M cherry, and we'll say snap gene reverse translated. Okay, cool. So it has this stop code on, and okay, so that's actually good. That's actually good to know. So I should be able to now go to this thing, gene fragments, order now, amino acid sequence, manually import. Got it. M cherry, codon usage table, E. coli. And then what we want to do here is we want to copy this thing. And did that have the... Okay, good. Continue. So now, okay, now we have the sequence here. Now we can download the sequence. Let's get the GenBank file. So we're going to go to downloads. And we're going to rename this mcherry twist reverse trans. And we're going to paste this into try2. Okay. So now I'm curious though. So they're both the same length, which is great. But I am curious to know if they're different. So, so let's see. So this is out of date, right? Can I refresh? Maybe it just no. It definitely shows. Okay, so how do I read GenBank and .dna files in Julia? I think the .dna might be let's see. This sounds like it's from forever ago. Wow, it just like totally made that up. So you have the GenBank, but I guess we... Let's see, what did it say about... It would actually be sick if Python call used UV as its environment, man. That would be dope. Okay, in any case, what we'll do is we'll just export them all to GenBank. So we'll add this. Wait, why am I on the wrong environment, bro? Actually, let's rename this to standard. That's what it's called, right? This thing had a random dependency on data frames, but you know, who cares? Oh yeah. Okay, so let's do the export then. The exports. So what do we want to do? We want to export this thing. Batch convert file format. Please choose files to convert. We want to convert this, and actually wait, we only need to convert this because because we're just trying to compare them. What is the difference between these? Okay, wait, convert. What just happened? Okay, here we go. So, but actually what I want to know is well, one thing that we could do is we could actually just do, this is kind of the simplest thing, is we take mcherry reverse translated. Where's the twist one? Then the twist one. And then we compare selected. Yeah. Isn't that wonderful? So that's not going to work. This file was created by twist by exported snapdrain. Okay. Okay, so we need to figure out what is the common way to diff. What's the common way, either on, you know. Oh shit, I had chat messages, bro? No freaking way. Bioalignments.jl. Dude, I never get chat messages. This is my first time getting chat messages in so long. Uh, Yeah, I read the chat. Sorry, guys. All right. Oligo, literally my name. No regrets. What do you mean you have incels, but not the... Wait, when did I say incels? Good brother. Are you a student? What are you ordering reagents for, man? Bro, I literally spent an hour explaining that. What's this about? Bro, I literally spent an hour explaining that. What's your job? Programming and biology? Does he even read chat? All right. Good stuff. Needle water? Okay, what is needle water? BioPython. Bio alignments. Biosequences. Biosymbols. Memory views. Biostructures. Biosequences. Is there really an alignment thing? Okay, there is a real alignment thing. Can we look at it? So does it render a picture like this? Because that would be pretty sick, actually. Pairwise. Holy cow. Come on. Just give me two sequences where I can look at it. I will be reading chat from now on. If anyone is still here. All right. Wait, can we go back to this thing? Oh, sick. Okay. So wait. But I guess we need to read these in to get the text of them. How do I read the file? Genomic annotations.jl. Okay, here we go. So this is going to be seeks try to, and then what is this one called? This is going to be called this. So this is going to be snap gene. Twist is going to be the same thing, except this name is going to be this. Okay. So now what we want to do is, I guess we want to copy those to, okay, oh my god, bro. Julia is such a joke of a language. Shit never works, dude. I think what happened was it started in the wrong environment. I don't know. But yeah, this thing is a total joke. All right. But basically we just need to get the sequence, copy it, and then put it into this emboss thing. Interesting. Okay, so we definitely want needle. If I install emboss, oh, that's cool. Is it Python? I don't know. Um, all right. Wait, I didn't answer. What was my question? What is my job? Bro, my job is being fucking chill like that. Bilal, come on. For real? My job is being chill like that, bro. Paralign, global alignment, affine gap score model. There's a lot of knobs on these algorithms. Let's just okay, let's just do this. Okay. What was the diff here? I want to see what the diff was. Oh yeah, but the thing is, these dot. It's so annoying that these. It's actually way better to export to GenBank if you're gonna be looking at it in text, because otherwise, it's a binary format. And it's like, what the hell am I supposed to do with a binary format? Also, it seems like there's. I don't know. Maybe it's for performance. I don't know. It just. I'd be interested to hear why they decided to make their DNA format binary, or like, some non-ASCII whatever. Come on, buddy. I feel like there's no reason for this thing to. Whatever. It's just so slow. Okay, cool. Okay. Wait, what happened? Wait, Seeks try to M cherry twist. M cherry twist rev trans. Oh, it's called .gb. Okay. What happened? What? Twist gave me a malformed. Wait, did Twist really give me a malformed GenBank file? Wait, oh, I think it's probably this. Wait, what? Couldn't parse location description. Okay, wait, hold on. Because, like, what is the error? Parse feature? Honestly, wait, can we look at the other one? 1.711. I feel like it's the same thing. Location qualifiers. Organism M cherry. Okay. Dude, I don't think... Bro, what? I don't think genbank.jl is even a real thing. Interesting. So the location starts with leading spaces? Ay, ay, ay. Okay, let's see if biosequences can read this thing. It's BioPython, right? Okay. Oops. Okay, so I forget what all that is, but I don't really care. Okay, so let's just do this. Ooh, that's nice. Okay, so what is it? Seekio dot read file name genbank. And it needs to be quotes. Quotes. Okay. Okay, so let's oh, is that wait, actually I think that one sucks though. Okay, also didn't even work. So wait, if I close this thing, then I do this. Bro. Did they really? Come on. Wait, what's happening? It used to actually work though. They did some stupid update and now it's broken. Bro, these software engineers are so stupid, man. I should downgrade. Why isn't it working? What is this? What is even happening? I mean, what? They broke the fucking thing, dude. Wait, is it actually? I think it's working. It's just like they have some weird thing to try to redirect this standard out to that weird thing. Did it actually tell me to Actually, wait. Let's see. Does it work? It doesn't send. Wait, hold on. No, that's stupid, man. Come on. Okay. In any case, so now if I do SG, okay, so this is Snapgene. Oh wait, actually wait, hold on. What am I doing, dude? Sometimes I do the stupidest stuff. Okay, so Snapgene will be first. And twist will be second. Okay. Pair, sure. Submit twist versus Snapgene. All right, let's see. Oh wow, they're actually not nearly. I was expecting them to be like very similar. Tool output download. What's that? Oh yes, a .out file. Isn't that nice? Uh, but they don't give you like. What? Oh, it wraps it. That's silly. Okay, I'm fine with that. But that's funny. Um, wait, this is okay. Do we have twist? Twist dot seek dot lower print. They are pretty different. I was kind of not expecting that. Cause wait, did it give me amino acid identity? I didn't even see that in there. Okay. Um. So let's So we should probably prefer the twist. Um. How do I get this emboss tool? That's not a real thing. What the? Oh wait. Bro, come on. I'll post an issue. I don't. Because Python just sucks. I hate Python so much. Do I actually want to spend the time to do this? I think I'm gonna put on some pants, do a walk around the block. We'll come back, figure out, because we're still in the stage of we're trying to design the insert, because we got the Puck 19, we got the promoter sequence. So if we look here, we are basically going to copy this whole thing. Uh, okay. Okay. We're gonna copy this. Because I don't know what any of that is. Okay, all right. Well, whatever. Um, so we should probably prefer. Well, yeah, because uh, well, that's the thing because the flow is like first you. Okay, so like uh um first get the p-tac promoter from. Bro, why doesn't it? Can I get some water and put on some pants? But I'm assuming we should probably. Bro, it's insane. Like, I don't know. That's crazy to me that like at this point there's like Yeah, there's no way Twist provides that algorithm for, but that's interesting, you know? It's like it adds an extra step to the process. Okay, I'm gonna. All right. So, um, um, yeah, one thing that's weird about this tool. So, tools, design synthetic construct. So the weird thing here, insert feature, standard features, custom features. You know, it's like, why can't you just import? Yeah, why is that grayed out? What the hell? Insert bases. It just seems so weird to me that. Okay, wait. So what are we doing now? We're ready to do this thing, right? I don't even know why I'm. tripping up. So basically, we are going to take this thing and I guess I'm going to copy it, file, new, sequence, linear. This is going to be the file name. Yeah, sure. I'll keep the file names will grow and grow, but this will be ptac laco rbs. Okay, and then what we should be able to do is we copy this. So first, we insert bases, we paste this, insert, okay, and it keeps all of the things. Then we go to twist, reverse translate, sequence, get rid of these stupid sites, but we do want this. Okay, then we copy this, insert bases. Why is this red? Why didn't it maintain the... Oh, you know what? I think that's a dumb feature, man. So we're going to undo. We're going to edit. So we don't need the protein file, but we do want this sequence, this features control T. So this is going to be mcherry. And it's gene. Okay. And then we want the color, right? Oh, was that it? Here we go. Okay, good. Save that. Save. Okay, so wait, we actually want to... That's the problem, is keeping the gen banks in sync with the... But that's... But no matter! No matter! Okay, so now we just want to go back to this thing, copy this, paste it, insert. Now we have the mcherry as the thing. TGA, is that a stop thing? If I were to look at this, okay, that's a stop thing. Okay, so this is going to be ptac mcherry. Synthesize. Okay, good. So now what we want to do is we want to go back through this thing of... We want to do tools, no, actions, insert fragment. So we're going to get this puck19 thing. Oh, but one thing we need to do before we do this, we are going to, we're just going to annotate on top of this. We don't need the OG thingy. Oh, wow, this thing actually, when you download the snap gene, this has a whole bunch of primers in it. But we know what primers we want, so we're just going to do that. So basically, Control R, primer. How do I take the RC? Is this the RC? Oh, here. Okay. So we got this. So this is going to be inv rev. Okay. Then we're going to go here, copy this, Control R, paste it, reverse complement, in forward. Okay. So now we have this thing. What is this? PGEX? Wow, so these features just didn't get exported in the GenBank, but they're here. So this is cool. So so these are different by 1 degree, which is good. That's really close. So I'm happy with that. So now so these are our primers. So then we can save this. And when we okay. So all right, so we're ready to do this thing, I believe. We go hi-fi DNA assembly, insert fragment. Then we're gonna select this and shift click this. And you see here we got that, and then we got this, which is exactly what we said. Linears by PCR, that is what we want to do. Now here, we want to do PTAC M cherry, right? And we want to use this in this case because we don't have the overhangs. We want to use as a template for PCR. But the plan, so I thought it could just create the primers. Choose fragments that can be fused. So this is actually a great error because I thought this thing could come up with the primers to choose primers automatically. In the vector tab, select the site of insertion. In the fragment tab, select the region to be amplified. In the product tab, choose overlapping primers. Oh, nice. Feel like we want this set as default, and we want this to be like, maybe we should say like 19 to 30. The target TM for overlap of okay. So this is first. So this is uh chose oh, one chosen primaries. Okay. Oh, so it does have this Right. All right. We'll have to read the manual. Okay. But so if I add vector.4, but if we look at those primers, vector.4 and vector.rev, they're a little bigger. And what's their GC and CG percentage? Do they tell me that? 52, 60. The rev, which is the rev one? Wait, let's go back to the sequence. InRev has 58%. So I think I'm planning on keeping the okay, so this is going to be the left overhang. So we're going to do this. We're going to assemble. And that's exactly vector rev. And then this overhang is not quite the same. How far does this go? See, so the recommended overlap there. I just wanted to look at it. So, okay, so these are the chosen primers. So we can do this assembly, but what I want to do right now is I want to... Okay, so let's just assemble it. That's fine. But what I want to do is I want to do actions, tools. No, what is it called? Design construct. And here what we're going to do is we're going to take ptac mcherry, copy the whole thing, insert basis, this. Okay, then what are we adding? We are going to add... We are going to add just literally invrev to the beginning. Okay. What just happened? Oh, interesting. Okay. So I copied that. Okay, that's fine. Oh, you can't add a primer here. Okay, that's fine. Let's go back to this. Now we want to go here and we want to add this. But we want to add the... No, we want to add that. Wait. We want to add CGAC. Wait, I missed this. $20 base pairs. Oh, wait, no. Invrev. We wanted invrev. Okay. Okay. And then here, we want in forward. And we literally want in forward. I want to go here. Insert. And it didn't freaking do it. What? Okay, let's just do this. There we go. Okay. I actually like how it did this. So now this will be ptac m cherry overhangs. And we're literally using the same primer. Yeah, exactly. For the 5' overlap, wait, is there a way to copy double-stranded? Bro, I think it's, wait. I need to copy all these because I've done this so many freaking times at this point, and I just don't. Okay. Okay. Blum 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 blum. Okay, so wait, so we can finally go back to this thing. This is the designer. Okay, so we're good, right? We added GACAGG. We get the GC percentage and the GC percentage here. Right. Okay, cool. So now what I want to do is I want to go to any builder and try their tool. So we're going to do new fragment, vec, load file, select file. We want, I forget exactly what we want, but we want probably just puck 19. We just want plain old puck 19. Oh, they don't accept, okay. Okay, so what we're going to do is we are going to, you know what I want to do? I kind of want to, all right, let's just, okay, so we have this file, bash convert, choose files. We want to convert puck 19. And we want to convert chosen primers. No. Wait. Chosen primers? No, we want the primer overhangs. I guess we also want... But what did this one... This one is the assembled. So that's assembled. We don't really want that. And then we also want to convert PTAC M Cherry. Because PTAC M Cherry, if we want to have it do the primer design again, which we do. Snapgene, GenBank, convert files. Okay. So I should be able to select bio, seeks, try to, modified. Okay, so we have the PTAC M Cherry. So we want the puck 19. It's circular. It's puck 19, PCR, auto generate primers. Okay, so here's what we want. So we did this a number of times yesterday, but I should just be able to go in. Actually, it'd be cool if it searched to the GenBank feature, but it's not going to do that. So we want to go to puck 19, add gene. Then we want to take this thing as our forward primer. And then we want to take this thing as our reverse primer. Cool. I like this arrow, man. I love these diagrams, dude. Little diagrams just make it so much more helpful. Okay, good. So, new fragment, load file. Now we're going to try to load with the overhangs. And PTAC primer overhangs. So wait, we didn't convert. We didn't convert chosen primers, did we? Wait, but what did that even do? That's not right. So we just want this, I think. And so this is going to be directly synthesized. There is a discontinuity immediately downstream of Puck 19. Wait. Build setting. Oh, wait, actually, hold on. Build settings. Oh, okay. So it's not like it's like, yeah, okay. But the thing is, if I change the primer, because I'm wondering, like, if I do, let's see, this one is 21, but this one, if instead I did, if I added one more base, uh, how do I select the complement? I guess wherever you do it, you can just do the complement. In overhang min 20 min 61. It's kind of high. The thing that I'm wondering though is it didn't give me it didn't calculate seem to calculate um The overhang. Um, Unlike PCR, it's not really as important that TMs for the overhang pairs are matched, right? So I could just bump the left overhang by one base pair, by one NT, and even though it makes the TM 55 versus 61, or the TMs for the pair now 55 versus 61, as before was 55 versus 59, or something like that. Maybe I will add that extra nucleotide. Okay, so we're kind of chilling. Oh man. Okay, so then maybe we should just add that one base pair to the overhang. That sounds pretty good. Okay, so then, okay, so we are going to add, so we are going to edit this sequence, and we are going to add a C here, right? Actually, wait, can we just do it all over again? Let's just do it all over again. So actions, no, tools, design synthetic construct. So here we're going to take ptac mcherry, which is all good, paste this in. Oh, wait, insert base, paste, insert. Then we're going to go to the assembly with the chosen guys. Then we're going to go like this, and we're going to select this C. Okay, so we're just going to make it a little bit longer, because I don't think it matters. Ugh, why does it do that? I hate that it does that. There we go. Okay, now I want to copy this primer overhangs. I want to do this. I guess we need to close this one. Okay, so now we're still gonna have this thing, but but we should be fine. Okay. Alright, we did it. So all I want to do is I want to check on my primers tab. So wait, do we want to redo the assembly? Actually, can we redo the assembly? Does that work? I thought that maybe this one. Let's see. So first off, vector puck 19 linearized by PCR. We're getting good at this thing, man. Okay, create blunt ends. Choose fragment. Primer. Oh wait, is that what I called it? That's not right. I should not have done that. Oh shit. I really screwed that up. Primer extended overhang. Ay. Okay, puck 19. NEB hi-fi, insert fragment. Okay, linearized by PCR. InBrev, InFord. Fragment. Please choose. Uh, 23 mer. No, see, it started with a. Yeah, that's good. We wanted the 20. We added just like a couple of base pairs to both sides, and the used directly as the fragment. Ready. Oh yeah, baby. Puck 19. P-tac M cherry, overhang, direct fragment, assemble. Final. This is the one. So basically here in the history tab, this is basically the Oh, is there a way to actually print print history tree text? Let's see, print to PDF, print. Where did that thing go? Where did that thing go? Oh, okay. So it's just in home. Pretty cool. Is the only thing print to PDF, or can I get an image? Oh, nice. Where did it go? Is there not a refresh thing for this? I feel like this is not even right. Where did the PNG go? Oh, here it is. Why is this not? Oh, I'm just dumb. Can you save it as an SVG? Ooh. Oh, I like this. I like this a lot, dude. This is really, really neat. Because that is like what... Dude, how much is Snapgene? I actually want to I would definitely... like... I could... Oof! Okay, never mind. I thought it was gonna be like $50. Oh my god, can I get sponsored? Oh, okay. Even that's kind of high though. But that is nice. Did we get new chat? Yo, what's up? What's up, Ole? Bro, okay, we are locked in. I mean, this looks fancy, dude. Can we look at the SVG? Can we look at it somewhere that doesn't suck? Open with Firefox. All right. Sicko. Okay, so this is a look at this whole process. Okay. I might make a video. Let's see here. Okay, so this is a look at an image of about 30 hours, maybe more of work. What we're looking at is a procedure or a simulation of what's called a hi-fi DNA assembly, which is basically the process of taking a plasmid and a gene fragment that you order from a service, then linearizing this plasmid so that you cut out the LAC-Z gene, because I'm not going to need the LAC-Z gene. I should say that the goal is to insert this M-cherry gene into a plasmid that we can then insert into E. coli. Then we'll have E. coli that glow red. Now, the way that this works is you basically design a set of primers. They're called inverse primers, really, because you're basically doing everything but this gene, whereas traditional PCR, you're doing a smaller subset of the thing to try to amplify a single gene. We're trying to amplify everything but a gene. So then we end up with this linear DNA fragment, which just has the ampicillin resistance and the origin of replication. Then we designed this. There's sort of two ways to do this procedure. One, you take your gene fragment and then you do another PCR step where you add overhangs, because the way that this hi-fi DNA assembly method works is that you have to have overhanging base pairs between these two fragments. Otherwise, they won't add up. And actually, you need to have overlaps here and overlaps here so that it recombines into a circle. Luckily, this software actually does check that for you and make sure that it'll all work. And so here I'm actually opting not to do a second PCR run on my fragment to add the overhangs, but because it's only like 7 cents a base pair, I'm just going to save the PCR step and directly synthesize this DNA and just have it ordered so that I can then do hi-fi assembly on it. I haven't posted in so long, and I've kind of been wanting to. Okay. I'm a post it, bro. Wait, what the hell? Oh yeah, wait, continue. Oh shit, I'm on the wrong account. Fuck. God damn it. Oh, it's gotta reprocess. Come on. Come on. Oh no. Oh, I think it's gotta reprocess this one too. Oh, what the hell? It lost it because I can't... What? Oh, wait. Oh, I fucked this up. Oh, there we go. Okay. Okay. Damn, TikTok's the slowest one. What in the world? What in the world? Okay, so I'm pretty happy with this though. I really like this diagram. This is very exciting to me. That basically, oh, that's so sweet, dude. This diagram is so clear what's happening now that I understand it. Overlap and insert. Insert between DNA ends. Bro, I'm a freaking genetic engineer. Nobody can tell me differently. Rumspringer. Okay. Um. So now, what all we have to do is we have this final thing. We can close everything pretty much. Um. This. Oh, and I can click to it. Wait. If I click on that, oh, see, that's, oh, see, it didn't like update, man, because that's not what we want. We want extended overhangs. So what we're gonna do is we're gonna bulk export extended overhangs and final. All right. Optimize the overhang lengths to to comply with any builders 20 NT minimum. Basically, used the recommended overhangs produced from. Is that what we did? I think that's what we did. I didn't even pre-watch this, bro. I just posted it. 30 hours, maybe more of work. What we're looking at is a procedure or a simulation of what's called a hi-fi DNA assembly, which is basically the process of taking a plasmid and a gene fragment that you order from a service, then linearizing this plasmid so that you cut out the lax Z gene, because I'm not going to need the lax Z gene. I should say that the goal is to insert this M cherry gene into a plasmid that we can then insert into E. coli. Then we'll have E. coli that glow red. Now, the way that this works is you basically design a set of primers. They're called inverse primers, really, because you're basically doing everything but this gene, whereas traditional PCR, you're doing a smaller subset of the thing to try to amplify a single gene. We're trying to amplify everything but a gene. So then we end up with this linear DNA fragment, which just has the ampicillin resistance and the origin of replication. Then we designed this. There's sort of two ways to do this procedure. One, you take your gene fragment and then you do another PCR step where you add overhangs, because the way that this hi-fi DNA assembly method works is that you have to have overhanging base pairs between these two fragments, otherwise they won't add up. And actually, you need to have overlaps here and overlaps here so that it recombines into a circle. Luckily, this software actually does check that for you and make sure that it'll hold. And so here I'm actually opting not to do a second PCR run on my fragment to add the overhangs, but because it's only like 7 cents a base pair, I'm just going to save the PCR step and directly synthesize this DNA and just have it ordered so that I can. The thing is, I actually think that one's wrong. I'm pretty sure if we look at the sequence of this, I'm pretty sure it's going to be wrong. Because. Well, it's. No, it's not going to be wrong because. It's just this history thing is wrong. Like, I wish I could change this history. Okay, I'll be right back. Okay. Um. Wow, I've only been streaming through 3 hours. It's kind of crazy. It feels like longer. All right, so we should get to the bill of materials thing. I think I. Okay. Um. All right. Do I have a? Let's see. Actually, let's see what happens if I do sheets done now. Okay. This works for me. Okay, so we're gonna do name, cost, volume, storage. Oh, wait. Shelf life, manufacturer ID. Manufacturer. Because you know I can spell. Bro, they always give you this crap, dude. I'll give you this crap. Okay, so let's just put some of this in here. I wonder if it can actually just populate this for me. Here we go. Here's the master bomb. Where do they sell TE? They sell TE buffer? No. Where do I buy TE buffer? Bro, come on. What the fuck? That's insane. Okay, so um um okay. So let's do this. Get rid of this. Get rid of this. Get rid of this. I would like to optimize that thing, man. That gene fragment. Okay. Okay, here we go. PCR master mix. Q. Okay. There's a lot of options there. Yes, the primer. So the inverse primers. For now, I think I'm just going to get the inverse primers. I don't want to go like overboard and buy more than I need to. But I think there is some more stuff to do in there. Okay, here we go. TAE buffer. Safe gel stain. Gel red. Sibber safe. Yes. Safe ladder. 1 KB plus. This out of things that I'm probably gonna buy, I feel like I might buy my own ladder. Do I need loading die? Because what is happening? I take the. How does this all work? Okay, so we are gonna need loading die. Okay, good. Did I? Yeah, I wrote that down. Okay. So you mix PCR product with loading die. That okay. Okay. So we're gonna need a loading die. Okay, so we got the ladder. Okay, good. So we got the ladder, the loading die. We don't need the extraction kit. Okay, we need the DPN1. Cut cut smart buffer. PCR cleanup, yeah, we need the Monarch PCR cleanup kit. Okay, yeah, gene fragment. More TE buffer. Oh yeah, wait, what about the IPTG? All right, so let's go through it. So this is gonna be insane. I'm not going for the name brand shit, bro. I trust 500 milliliters. Actually, we should just move URL. To here. Or actually, we should move it to here. I'm gonna go for the. I do want it to say sterile at the very least. Okay. The ethanol. Can we move this down? I don't know about the ethanol. Okay. Q5 master mix. This shit is so expensive, man. 100 reactions. Or wait, that just dude. Oh, what? Bro, how do I fucking How do I... I hate this. Okay, whatever. Okay, well, we can do that. That's fine. Then we should be able to say... Okay, do I have a... I don't think I have an IDT login, so we need to make an IDT login. And I'll show you guys how that works. Okay. Oh, here, wait, I have to do this way. Okay. All right. So we're here. So, okay, so this is what you need to do. If you want primers, you have products, oligos, custom DNA oligos, single-stranded oligos, and then you're finally ready to do this thing. And now we want to go to SnapGene. This is the final map, right? So we want to go in here to the sequence. We want this. This is going to be called, so we have 2 items. This is going to be called invrev. I'm going to paste this in here. Okay, go. Then we're going to do inv forward. We're going to go to this. Go down here and we're going to copy inv forward. Okay. And the TMs are really close together. They're pretty close together. And they're both within the CG thing. So, Primer 3 did a pretty good job. The only thing that now I'm wondering is like, I probably don't need anything. Okay, good. Okay, so then we go add to order. Check out. Um, I'll fill this out. I should just memorize it. It was close. Bro. Oh. Okay, so the primers 43.92. Would they say 12 nanomoles? Okay, but we got the price. Now TAE buffer. Okay, the gel stain. So the thing about the Sibber safe is that they make you buy an obscene 400 microliters of it for $95. So what I'm sort of inclined to do is buy gel red, which is this. And I hate that it does that. Okay, so it's 10,000 X in water, 3X in water, and 10KX in DMSO. Bro, my shorts get no views. I don't know why though. It used to be like a flat 500. Now it's like nothing. Okay. Totally fine electroporate. You don't need chemically competent. Yeah, I'll just make them chemically competent. Okay. Um, insert column left. That's not what I meant. I don't know what left and right is. Notes. Can we hide this thing? Okay, let's just do this. Wait, what just happened? Are you joking? Okay. This is going to be gel red 10kx in water. And it is how much? Okay, we want the trial size, so this is going to be the 0.1 milliliters, and it is $34. Okay, safe ladder. Wait, what happened? $87. Um, I don't even know how many that is. That's like 125 lanes. That's not even that many lanes, dude. Oh, actually, that's a lot of lanes. Never mind. I'm lying. That's a lot of lanes because this is the ladder. This isn't the loading die. Okay, so that's what we're gonna get. No SDS. Okay, wait. Oh, I've been forgetting to oh, yeah. Broche. Oh, it only runs over if there's not a cell. Okay, that's nice. Okay, it's this 6x. Okay, this is the volume is 4 milliliters, and it is $52. Okay, DPN1. Dipney. Okay, so when I said $500 reagents, I meant it. Okay. Or actually, wait. Can we just upload a gen bank? FASTA. Bro, what the hell? What are you talking about? Name is too long. Okay. Oh, it's because the... Okay. All right. So it was like 58. Okay, so 58 no shipping. And the total... I don't know why I'm writing that down because I have to pay for shipping. Bro, this shit sucks, man. This shit sucks, dude. Oh my god. How many was it? 829? All right, so we have that quote, so we're good. So let's get back to it, back to work. So hi-fi master mix. This stuff is expensive, man. So we can get the hi-fi master mix, or we can get the kit. Can we actually look at the phenotype of this thing? Or wait, actually, sorry, genotype. See, this thing doesn't have that lack eye thing, so we wouldn't even be able to use this. Say what? All right. So do we even want this? I feel like I just want the master mix. I don't even want all this. Wait, but what else did it come with actually? It came with some it came with 25 mils of SOC. I mean, it's insane. This stuff is so expensive. 100 microliters is $180. God. So this is like $25. So you kind of do make, but what am I gonna do with SOC, bro? How did they ship this? They shipped this
For decades, the protocol begins with insert DNA and a cloning vector. The insert DNA can be an individual fragment that has been amplified from a source DNA, two annealed oligos, or a restriction fragment from a vector. Cloning vectors are identifiable by the presence of a multiple cloning site, or MCS. Restriction enzymes are then used to digest the insert and vector DNA to create compatible Ms. Next, these parts are assembled using DNA ligase to form a covalently sealed plasmid. Finally, the ligation reaction is transformed into competent E. coli and plated to identify successful transformants.
nice. I guess let's just get the kit. Who the fuck cares, man? All right, let's get the kit. It's 212. Because then we have, I don't know. We have another strain, I guess, which is nice. Straight from the source. So we have 10 things of 50 microliters? Really? Okay. All right, let's look up. Do they sell IPTG? Wait, actually, let's look on Amazon. Amazon is where I want to go. I don't really trust this, though. I don't know. Is this a legit company? Seems kind of sus, man. All right, what about yeah, this shit sucks, dude. 1 gram. Who sells this? I don't want the powder. Oh, 50 milliliters. How much is the salt? So wait, if we want 100 milligrams per milliliter, and we have how many? 250 milligrams. Oh, really? It's way cheaper to make your own, actually. Because I can make 150. Oh, yeah. Wait, yeah, there is a math slip. What the? Wait, really? Wait. Why is that so much more expensive? That's insane, dude. That actually makes the that actually makes the liquid way more no, leave out the ethanol. Okay, wait. I don't want the quick load stuff. What happens if I just paste this into Discord? How do I hide this thing? You can't hide this thing? That's crazy. I feel like you should be able to hide this thing. Oh yeah, you can.